Hi, the question for this video will be, can we do differential expression analysis with two factors, where one is continuous like age and the other is categorical like gender? And this is not exotic question, it's very common for human studies. For instance, take this family. The minimal description of the samples taken from these members will be age and gender, and pets will be excluded, obviously. Right. I cannot say it is a usual choice, especially for the aging studies. Quite often ages are replaced by categories, young and old for instance. And this is unfortunate because it will create a gap in your age scale, and then it will complicate your trend analysis if you will ever want it. Previously I made this video where I showed how to use absolute time scale in edgeR protocol, and it was a one factor experiment, so only age, nothing else. This story was based on this chapter of edgeR tutorial, and the trick was in using polynomial regression, and the way how you do it, you put a line above your design where you define x as a derivative of a time and then put this x into your model matrix. Now the question is, if we use time transformed by this poly function and gender, will it work in edgeR? For this exercise I decided to use this GSA dataset which I used before. That's the number and that's the description. It's a human aging, about hematopoietic cells, two age group, young and aged, also known as old. It's not ideal, but it should work. This dataset was previously used in this script number 5. For this video I wrote another script where I simplified the beginning, so I load the libraries I previously processed and saved, counts table and sample tables. I just open it and use. As I said, the middle ages are missing. Ages are split in two groups, young and old. With this we declare DGA list. Then we check samples, and then we see that indeed we have groups, young and old, equal by size, but pretty biased by gender, unfortunately. And age column is also present. Then, as usual, we filter by expression. Then I like this palette, let's use it. This is how data look before normalization. Then we normalize, very nice. MDS doesn't show any outliers. Then we simplify the row names. We will need it for merging with annotation. These decimals will be removed. Then we extract immediately normalized data as log CPM observed, like this. And then we simplify the row names. So now we can do option 1 with the age groups as before, and then we can do age column, that's the new one, and then introduce design. X will go for polynomial transformation of time, and add this X. Will it work or will it not, and how? So let's take the first design. Then we can check what it is, nothing unusual. Then we estimate dispersion, and then we go with the fit and fit design and the contrasts. Yeah. Then we get some differentially expressed genes, and then go to the results table and tops. Then we have to annotate this stuff and merge, and then it will be group tops in this case. Now we repeat design with the poly and age and sex. Then we check it. So the design is different. We have intercept 3 columns of the polynomial function, and sex is also present. Then we estimate dispersion, so the same plot. Then we do a fit. Fit takes a new design as well. Then we cannot do make contrast because there is no contrast. We don't declare it at all. Instead we say coefficients for the table. It means these columns 2, 3 and 4. That's what we fit for. And then as usual TT and tops and annotate and merged, and it will be H tops. Good. Now we call the Euler and compare ages and groups with each other and see that our group analysis reveals nearly 600 differentially expressed genes, but absolute age analysis give you much smaller numbers. Well, first of all, the algorithm is different, and secondly, absolute ages scale assumes some reasonable trend observed in your data, and this limits the number of the differentially expressed genes. When you do differential expression analysis with the continuous variables, you have to keep in mind the model of the linear regression where you in the input you have some categorical or continuous variable, and in the output you generate a fitted values. The same way works in a differential expression with ages. So we extract the fitted values, which we'll compare with observed, and in a regular regression model you can visualize your observed and predicted values in one plot. Dots are observed and line is predicted. And in the real study you will have to work a little harder to visualize your data and results. We create a subset of CPMs observed and fitted for 40 genes found in H tops. Then I extract ages and sex again from the Y table and bind it to the end of those two tables, Y. That's my way of ordering the columns by age and coloring by sex. So it is a little bit unusual style that here are the genes, here are the columns, but at the end I have ages, which is now in ascending order, and the sex 1 or 2, which I can use to discriminate between male female. Yeah, order them. And now time to illustrate. As I said before, the problem if you use absolute ages, how you supposed to illustrate your data. So let's say take gene number 1, and then we plot observed ages and color it by sex. Yeah, ages is the X scale and E is the line for particular gene over there. And this is the gene name. And next we show dots for observed values with the colors of sexes. And next we add trend lines which are based on fit values in separate for male and female. It looks like sex has an effect on expression of this particular gene. And if you have number 2, you can repeat it. And then you see that in this particular case, sex doesn't have much effect on the trend. but at the same time you realize that sex distribution in age groups is so terribly biased that, let me see again. So there is only one male in the old group. Here it is. Yeah, blue is usually male. And there are 3 females in the young. It's a bit shaky, but for the illustration purposes it is good enough. This is one way of showing your results and trends, and you can multiply it for 9 or more genes in your list. But then of course you can say, can I get all 40 genes together? And the answer yes. Then you use library tmixclust, which I also explained you in a previous video. This tutorial you can find in Bioconductor. And the major purpose of this is the clustering of a time trends. In principle, you can use a function plot time series df out of this library directly using all 40 genes which you have in your final group. You declare input data frame and the time points, and then you plot them all. But then you will see that it's quite a mess, and therefore you better do it more slowly, first split on clusters. We use timescale, which is hidden in our CPM fit table, and for expression values we use the same table truncated, remove sex and age, and then we say give me 4 clusters. It says okay, that's the groups. Then we get cluster assignments as a color and plot MDS. MDS is based on Euclidean distance, whereas better ways use correlation distance. But even with Euclidean distance you see these 4 clusters split in clouds, okay. And then we want to show, for instance, 1 cluster out of them, which is number 2, that's cluster 2, okay. The disadvantage of this visualization that you can do only 1 in a time. Number 3, let's put them all together. Here I go back to the regular plot function and do some extra work to split data on genders. I do not draw lines, and these are not multiple lines but only one. It is the column means for each cluster. Now we have a n time and the gender at the same time, and if you plot everything it will be a bloody mess. And therefore, in order to make it somewhat cleaner, you project only mean values for each cluster, and then give me a color and a symbol different for male female. This dataset reached its limits, so there are missing middle ages and sex distribution is problematic. In principle, you can also draw, let's say, trend lines for one and for the another group, but it would make all sense in case of a more powerful data. Our last final results table is merged CPM, where you combine expressions and gene names, and at the end the cluster name, where you can recreate it with different algorithms. And this merged CPM and merged are your end results in this particular analysis. That's how it works. So the answer to the question, would it work, is yes.